Okay, my memory stick ran out. It's a delete file, so that's why this is already here. So different types of mutations, you get point mutations, which are the least bad, and then substitutions, additions and deletions are the most severe. Okay. So point mutation is caused by a change in one base pair of DNA or a single gene. So a gene has one base pair and that gets changed. So caused by a change in one base pair of a single gene. Okay? So it occurs in a gene, by the way, which just basically equals a protein of DNA. Okay? It can also actually include RNA as well, but for this course we're going to deal with DNA only, okay? Or it can be a single or it can be a one base pair change in some other part of the DNA that is not actually the gene, which isn't actually the gene. Okay, so these they basically cause change to the gene expression. So an example, if you have point mutations, so DNA, okay, you have a bit of DNA like this, okay, with that, another region here, okay, so this bit controls regulation. So this is a regulation section. So this is unaltered. Okay. This then leads to, you get transcription, translation, and trans. Okay, so this leads to a normal protein. Okay, and a normal phenotype. Okay, so this is the regulation. This is a regulatory region that has an effect on the gene. Okay, both are unaltered. Introduce point mutations. So this is actually a situation where there's no point mutations. Okay, so in this region, no point mutation. Now if we introduce so that's DNA. Okay. And then introduce here. Okay. So that is a point mutation. This is the regulation part again. Okay. And that's the gene. Okay, so this has an effect on that. this part is unaltered, and this is altered, okay? So this results in again you get transcription and then translation, okay? Transcription, translation, that's just abbreviations for transcription, translation. Because the point mutation is in the gene and that's being expressed, you get an altered protein. So you get abnormal protein, okay, which is non-functional. It's non-functional protein that's synthesized. Okay, and the phenotype is altered. Okay, and then you get another example where you get point mutations, where you get DNA. Okay, so you get like that. You get the regulatory region here in the gene. Okay, but in this case you get a point mutation in the gene. Okay, so point mutation, and that's the regulatory region. Okay, so in this case, the normal, you don't get any, this does not have an effect in that, so therefore you get no transcription and no translation and no protein. Okay, so phenotype. Okay, so because this is changed, this cannot express the gene, so you get nothing happening. Okay. Okay, so that's point mutations. Next type of change is there's also substitutions. So substitution. Okay, so you get DNA. Okay, so if you get a normal DNA, a mutant, okay, so you get G, G, C, C, T, C, C, T, C. This leads to an RNA, which is C, C, G, G, A, G, G, A. Okay, A, G. Okay, so, and these amino acids, these are bases, so that's C, C, G, that's proline. GAG that's glutamic acid. That's glutamic acid, and GAG is also glutamic acid. Okay, but when there's a substitution, if you write this as G, C, C. So in this case you've replaced the T with A, okay, and then C, C, T, C. So this results in the messenger RNA being C, 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 G, G, U, G, G, A, G, okay? So we just divide off into the codons. So you get CCG is again proline, but GUG is not, GUG now exists. Have I done that correct? GUG now exists, which is actually valine, and GAG, which is glutamine. Okay? So the glutamic acid, sorry, the glutamic acid here is actually being replaced by valine. That's glutamic acid. The glutamic acid here, which we should have, has now become a valine, okay? So this is a major structural change. So this is actually hemoglobin S form, okay? So a change in one amino acid causes sickle. This is actually what occurs in sickle cell anemia. So change in one amino acid, change in one it causes the red blood cells to become sickle shaped, So this is the cause of sickle cell anemia. And how do you spell anemia? A-N-A-E, sickle cell anemia. The red blood cells become sickle shaped. Okay, it's homozygous So when people are homozygous for it, meaning they have the two recessives, they suffer from sickle cell anemia. Okay, so that's substitution. The T is replaced by A, so T becomes A. Okay, I'll make it clear that T there becomes A there. So that's an example of substitutions. Then you've got insertions. So DNA, you got A, G, A, G, T, C, T, T, C. Okay. I should have actually start again. Actually, too much, not enough space. So you got A, G, A, G, G, T, C, T, T, C, okay? So the DNA, that leads to RNA, which is U, C, U, C, A, G, A, G, okay? But that's normal, and in the mutated version, you got A, G, A, okay? Then you got, what was that meant to be? That's meant to be a G, that's a G, so you got G. But we've also added another G, but put it in red. Then T, C, T, T, C, okay? So AGA, GG, TC, TT, okay? So that becomes U, C, U, C, C A G A A, okay? And so when you make the amino acids of these, U C U is that's a serine, CAG is glutamine, okay, and AGG is lysine. When you make the codons of this, okay, that's a serine, but because you've added a base, okay, so G added, this now becomes the glutamine becomes a proline, okay, and the lysine becomes a glutamic acid. So you get this major frame shift. Everything after everything after serine, all the amino acids after the serine are completely different. Okay, so this is a frame shift. It's completely different. It's altered. Everything's altered because of one. Because you've added a single base in here. It means the whole meaning of each codon is different. So if you simply do a substitution, the codon would remain the same position. Because you actually added one, everything else gets shifted along one. So this is our frame shift. Okay. And lastly, when you remove a base, quickly go over that. So DNA, you have A, G, A, G, T, C, T, T, C. That's normal, and this is mutant, okay? A, G, C, T, T. C, G, okay? So in this case, that first T there, after the A, G, have I done this right? Yeah, that T, in this case, is removed. Okay? So after G, you now get C, T, T, C. So this whole thing, all the bases, all the millions of bases going that would get shifted back one. So this is the frame shift again, and it messes everything up. So you get RNA formed, which is U, C, U, C, A, G, and the RNA here, which is U, C, U, C, UCU, C, G, A, G, C, okay? So here, and here, remember these three are from the mutant and these three are from the normal, so that makes, that's a serine, CAG that's a glutamine, and AAG is lysine. But now, in this case, that's still serine. Because of this frame shift, CAG has become CGA, so it's become arginine. So the glutamine has become an arginine. Okay? And the serine, which was this, sorry, that was lysine, AAG, that's now become a serine. So the serine remains serine, that glutamine remains, yeah, the glutamine becomes an arginine, lysine becomes a serine. So everything's frame shifted. So because of one amino acid is deleted, all other amino acids after serine are changed. So this is a frame shift. Okay? Yeah, hope that's clear. All amino acids after this have been changed because one's removed, suddenly they all become shifted that way, so the codons become different. So these are major, deletion, insertion, and cause major changes to the proteins. I think I said earlier on that the most severe, certainly additions and deletions are the most severe. Substitutions can be bad as well. They can alter things, but these are actually the addition and these two are actually worse. They're most severe. Okay? Thanks. Good luck with your studies in this. Any questions, ask me. This is our high grade.